The next topic is carrier-mediated transport. Carrier are proteins. Proteins are located in your cell membrane. The carrier is going to show specificity, meaning that it's going to carry only one thing. For example, the carrier for insulin is only for insulin. It's not going to be used by any other molecule. And saturation means that if the carrier can transport only 2 insulins, then if you have 5, it doesn't matter. It will transfer only 2 because it gets saturated. We scroll down and we have that there are 3 different kinds of carriers. There are going to be uniports, symports, and antiports. Uniport transports only one substance across the membrane. Symport transports 2 different ones, but they are transported in the same direction, and therefore that process is going to be called transport. Antiport transports 2 things, but these 2 things are going to be transported into different directions, meaning one outside the cell and the other one inside the cell. That process is going to be called a counter-transport. Achieved by these different type of carriers are going to be facilitated diffusion, primary active transport, and secondary active transport. In facilitated diffusion, the substances go from high to low. But it doesn't do it alone. It needs a transporter. For that reason, this is going to be called facilitated diffusion. The primary active transport is an active transportation because it uses ATP. Remember, we always say from high to low. Well, a lot of circumstances is going to be done from low to high. But that is only if we are going to use ATP. Secondary active transport, in this case, it will use 2 transporters. Here you can see you have one transporter, another transporter. You transported all these things inside. Now from inside, you're going to move it outside through these other transporters. Very important is the sodium-potassium pump. It is a very important transporter because the sodium-potassium pump maintains the membrane potential, regulation of cell volume, heat production. After that, we have vesicular transport. Vesicular transport is used for the transportation of large amounts of substances. In the case of this type of transportation, you're going to have 2 very important concepts. One of them is going to be endocytosis, when you put things inside the cell, like in this case, you see they are outside and they are being sent inside. The other concept is going to be exocytosis, where they are inside, the vesicle opens, the substances get outside. Examples of endocytosis are going to be receptor-mediated endocytosis, which are these ones are the receptors. You see the molecules are going to use these receptors in order to get inside. Another one is pinocytosis, where these molecules right here, they are going to be extracellular fluid. So pine is basically for water. Another important example is going to be phagocytosis. The cells, especially those cells that are going to defend our body, are going to get this, let's say, brown bacteria. Then our cell is going to form a vesicle like this, and then the bacteria ends up inside that cell that eventually is going to destroy this bacteria. Okay, the next topic is the cell interior. The first thing we have in there is the cytoskeleton. Cells look like cubes, columns, because the cytoskeleton is these structures right here that are going to give the cell the shape that they have. What are these cytoskeleton composed of structures called microfilaments, intermediate filaments, and microtubules. They are made of proteins. In addition to that, what else we have inside a cell? Then we have cell organelles. So this is the cell, and you have a nucleus. The nucleus is where the genetic material is located, such as the DNA. Other structures that you have is the rough endoplasmic reticulum, which are like tubes that have ribosomes on top of them, and they are going to be related to the synthesis and transportation of proteins. We have a smooth endoplasmic reticulum that are going to be related to lipid synthesis as well as detoxification. Ribosomes, they are going to produce the proteins. Golgi complex, they modify the structures that the proteins are going to have in order to release them. Lysosomes, that's where as I mentioned, for example, bacteria get destroyed. Peroxisomes, they're going to have chemicals to detoxify the cell. And mitochondria, where we are going to produce ATP. The centros are going to produce the mitotic spindle necessary for the cell division, and that's about it for this part.